Welcome everyone. As future biologists, it's vital for you to grasp how structure defines function in immunity. Today's class will give you a clear picture of how different immune organs support the formation and activation of specialized immune cells and how each cell type from neutrophils to dendritic cells contribute to our body's surveillance and defense strategies. I encourage you to think of these cells as a well-organized army, each with a distinct role in safeguarding our internal environment. Dear students, today our topics for discussion is structural features, characteristics and functional role of immune organs and cells, a focus on neutrophils, eosinophils, basophils, mast cells and dendritic cells. So, we will cover the topics under the following learning objectives. Number 1 will be understand the structural and functional organization of the immune system. Number 2, describe the structure and functions of primary lymphoid organs. Number 3, differentiate the structure and immune roles of secondary lymphoid organs. Identify the structure and functional specialization of major immune cells. Number 5, evaluate the coordinated function of immune organs and cells in host defense. The immune system is a complex network of cells and organs that work together to defend our body against infections and diseases. The immune system consists of many organs and tissues that are found throughout the body. These organs can be classified functionally into two main groups. Number 1, primary lymphoid organs. Number 2, secondary lymphoid organs. Primary lymphoid organs are the foundational sites where immune cells, especially lymphocytes, are born and trained before they head out to defend the body. These organs provide appropriate microenvironment for the origin, development and maturation of lymphocytes. Primary lymphoid organs include the bone marrow and thymus. First, we will deal with bone marrow. Bone marrow is the soft, highly vascular and flexible connective tissue within bone cavities, which serve as the primary site of hematopoiesis, that means blood formation. The bone marrow is composed of both cellular and non-cellular components and can be structurally divided into vascular and non-vascular regions. The non-vascular section of bone marrow is composed of hematopoietic stem cells of various lineages and maturity, packed between fat cells, thin band of bony tissue, trabeculae, collagen fibers, fibroblasts and dendritic cells. This is where hematopoiesis takes place. The vascular section contains blood vessels that supply the bone with nutrients and transport blood stem cells and form mature blood cells into circulation. Now, functions of bone marrow. Bone marrow is responsible for producing all types of blood cells, whether it be RBCs, WBCs, platelets, through the process called hematopoiesis. The bone marrow gives rise to all of the lymphoid cells that migrate to the thymus and mature into T cells, as well as to the major population of conventional B cells. B cells mature in the bone marrow and undergoes selection for non-self before making their way to the peripheral lymphoid tissue. Yellow bone marrow is actively involved in lipid storage, which can be used for energy and other functions. In adults, red bone marrow is mostly found in flat bones, while yellow marrow dominates long bones. Now, second organ is thymus. The thymus is a flat bilobed organ situated above the heart. The thymus is the site of T cell maturation. Each lobe is surrounded by a capsule and is divided into lobules, which are separated from each other by strands of connective tissue called trabeculae. Each lobule is organized into two parts, that is the outer cortex, which is densely packed with immature T cells called thymocytes, whereas the inner medulla is sparsely populated with thymocytes. Both cortex and medulla of the thymus are crisscrossed by a three-dimensional stromal network composed of epithelial cells, dendritic cells and macrophages, which make up the framework of the organ and contribute to the growth and maturation of thymocytes. The thymus gland is a critical organ of the immune system as it serves as a defense mechanism against different pathogens, tumors and antigens. Now, functions of the thymus gland. The most important function of the thymus gland is to induce the development, activation and differentiation of T cells so that they can perform their function as mediators of cellular immunity. After their maturation, they enter the bloodstream and travel to lymph nodes and other organs in the lymphatic system, where they help the immune system fight diseases and infections. The gland also induces the release of cytokines that are essential for controlling the development of T cells through different stages. As an endocrine gland, the thymus is known to produce two hormones, thymosin and thymopoietin, which play a crucial role in the development and maturation of T cells. Now, secondary lymphoid organs. In addition to primary lymphoid organs, there are secondary lymphoid organs such as spleen and lymph nodes. These organs trap antigen from tissue or vascular spaces and sites where mature lymphocytes can interact effectively with that antigen. Blood vessels and lymphatic system connect these organs, uniting them into a functional whole. Now, lymph nodes. Lymph nodes are small bean-shaped organs that filter lymph and contain immune cells. Afferent lymphatic vessels carry lymph into the node, while efferent vessels carry it away. The lymph nodes composed of a medulla and a cortex, which is surrounded by a capsule of connective tissue. The cortex is composed of two main regions, the outer cortex and the paracortex. The outer cortex contain B cells organized into follicles which can develop germinal centers upon antigenic stimulation. The paracortex, on the other hand, is primarily populated by T cells which interact with dendritic cells and are associated with CCR7 chemokines which attract T cells and dendritic cells. Medulla, it is the innermost layer that contains large blood vessels, sinuses and medullary cords. Medullary cords contain plasma cells that secrete antibodies, B cells and macrophages. Now, functions of lymph nodes. Primary function is filtration. The primary role of lymph node is to filter the lymph fluid that circulates through them. Lymph nodes act as checkpoints, removing foreign particles, microbes, damaged cells and tumor cells from the lymph. Number 2, lymph nodes are crucial for initiating an immune response against trapped microbes and antigens. Within the lymph nodes, lymphocytes interact with antigen presenting cells such as dendritic cells to recognize and respond to specific antigens. And last, antibody production. B cells, when stimulated by antigens, multiply and differentiate within the lymph nodes and develop into antibody producing plasma cells. Now, spleen. The spleen is a feast sized organ just behind the stomach that collects antigens from the blood. It also collects and disposes of senescent red blood cells. Structurally, the spleen is enclosed by a capsule with projections, trabeculae, that create compartments. These compartments consist of two main types, a red pulp and white pulp. A red pulp. This area contains sinusoids, macrophages, red blood cells and some lymphocytes. The bulk of the spleen is composed of red pulp, which is site of blood cell disposal. Then white pulp. White pulp surrounds branches of splenic arteries, forming a periarteriolar lymphoid sheath, primarily populated by T lymphocytes. Primary lymphoid follicles rich in B cells and some containing germinal centers are attached to the PALS. Now, functions of spleen. The spleen performs several crucial immunological functions. 1. Blood filtration. It filters the blood by trapping blood-borne microbes and removing old or damaged RBCs, thereby helping to maintain blood composition and preventing infections. Its primary function is to filter blood and trap blood-borne antigens, allowing it to respond to systemic infections unlike lymph nodes which target local tissues. Second, production of lymphocytes. The spleen is a significant site for producing B and T lymphocytes, which are essential for identifying and neutralizing foreign invaders. Third is immune response activation. Upon detecting pathogens, the spleen produces antibodies and activates immune response, especially against polysaccharide antigens found on bacteria. Number 4, reservoir for blood. The spleen acts as a blood reservoir, releasing it into circulation during hemorrhage or increased physical activity. Now, let us discuss mucosal associated lymphoid tissue, that is MALT. Mucosal associated lymphoid tissue is located within the linings of the respiratory, digestive and genitourinary tracts. The cell aggregates, that is follicles and aggregates of lymphoid tissue, are principally built of B lymphocytes, while lymphocytes are placed in interfollicle areas. Depending upon their location, they can be subdivided into nasal associated lymphoid tissue, NALT, located in nasal passages, first line of defense against inhaled pathogens, absent in humans, present in rodents and other primates. Second, gut associated lymphoid tissue, that is GALT, which includes the tonsils, adenoids and appendix and specialized structures called Peyer's patches in the small intestines. They collect antigen from the epithelial surface of the gastrointestinal tract. Number 3, bronchus associated lymphoid tissue, that is BALT, located in the bronchial and lung tissue. BALT is involved in the immune response to inhaled pathogens in the respiratory system, only present in childhood. Genitourinary associated lymphoid tissue, that is GWALT. This tissue protects the urinary and reproductive tracts from infection. Now, function of this mucosal associated lymphoid tissue, that is MALT. MALT is a diffuse system of lymphoid cells and tissues that are strategically located in various parts of the body, including the gut, respiratory tract and genitourinary tract. MALT tissue constitutes the first line of defense against harmful factors, and its main function is to initiate local immunological response manifested with the production of secretory antibodies. Now, let us discuss about tonsils. Tonsils, they are small masses of lymphoid tissue in the pharynx, act as first line of defense against pathogens entering through the mouth or nose. They are crucial for building immunity, especially in children, and form a ring-like structure called Wilder's ring. While important for immune function, they can also be susceptible to infections and inflammations, leading to conditions like tonsillitis. Now, structure of tonsils. There are 3 types of tonsils. One, palatine tonsils, located on either side of the throat, easily visible when the mouth is open. Second, pharyngeal tonsils, also called adenoids, situated in the nasopharynx behind the nose. Third, lingual tonsils, found at the base of the tongue. Now, function of these tonsils. Number 1, immune response. Tonsils contain immune cells like lymphocytes and macrophages that help to identify and neutralize pathogens. Number 2, antigen sampling. They sample pathogens entering through the mouth and nose, triggering immune responses. Number 3, antibody production. B cells within the tonsils can produce antibodies to fight off infections. Number 4, first line of defense. Tonsils act as a barrier, trapping pathogens and preventing them from spreading deeper into the body. Now, let us discuss about appendix. The appendix, a small finger-like structure connected to cecum, the beginning of the large intestine, is located in the lower right abdomen. While traditionally considered as a vestigial organ, it is now understood to play a role in the immune system and gut health. Now, immune functions of appendix. Number 1, it is a reservoir of beneficial bacteria. The appendix can store good bacteria, especially during gut infections like diarrhea, helping to repopulate the intestine with beneficial microbes after the infection clears. Number 2, lymphoid tissue. The appendix is rich in lymphoid tissue containing immune cells like B cells, T cells, macrophages and natural killer cells, which are crucial for detecting and fighting off pathogens. Number 3, IgA antibody production. The appendix produces IgA antibodies which regulate the density and quality of gut flora, maintaining a healthy balance of bacteria in the gut. Now, Peyer's patches. Peyer's patches are small clusters of lymphatic tissue found in the wall of the small intestine. Specifically, they reside within the lamina propria and extend into the submucosa of the ileum. Peyer's patches act as the immune system's first line of defense against microbial and dietary antigens. They monitor intestinal microbial populations and can prevent the growth of pathogenic bacteria in the intestines. Now, what is the immunological function of Peyer patches? Their function is to absorb antigens from the intestinal lumen via endocytosis and transport them to initiate an immune response. These Peyer patches contain M cells, that is microfold cells that sample antigen from the intestinal lumen, and these antigens are transported to underlying dendritic cells and macrophages which process and present them to T and B lymphocytes. Upon antigen presentation, B and T cells in Peyer's patches become activated. B cells can undergo class switching, especially to IgA antibody, which is crucial for mucosal immunity. Activated B cells migrate to the lamina propria and differentiate into plasma cells that secrete IgA antibodies. Now, let us discuss the cells of the immune system. The immune system relies on a diverse array of cells to defend the body against pathogens. All these cells, whether RBCs, WBCs or platelets, they derive from the same progenitor or precursor cell, that is the hematopoietic stem cell in the bone marrow. These pluripotent cells divide to produce two more specialized types of stem cells, a common lymphoid progenitor that gives rise to the T and B lymphocytes responsible for adaptive immunity, and a common myeloid progenitor that gives rise to different types of erythrocytes, that is red blood cells that carry oxygen, and the megakaryocytes that produce platelets and are important in blood clotting. The common lymphoid progenitor gives rise to the lymphocytes. There are two major types of lymphocytes, B lymphocytes or B cells, which when activated differentiate into plasma cells that secrete antibodies, and T lymphocytes or T cells, of which there are two main classes. One class differentiates on activation into cytotoxic T cells, which kill cells infected with viruses, whereas the second class of T cells differentiates into cells that activate either cells such as B cells and macrophages. Now, these adaptive immune cells such as B cells and T cells develop more specific response and provide long-term immunity. The myeloid progenitor is the precursor of granulocytes, macrophages, dendritic cells and mast cells of the immune system. So, these cells can be broadly categorized into innate and adaptive immune cells. Innate immune cells like granulocytes, megakaryocytes, natural killers, dendritic cells and mast cells provide the first line of defense. Now, let us discuss granulocytes. The granulocytes are so called because they have densely staining granules in their cytoplasm. They are also sometimes called polymorphonuclear leukocytes because of their oddly shaped nuclei. There are three types of granulocytes, all of which are relatively short-lived and are produced in increased numbers during immune responses when they leave the blood to migrate the sites of infection or inflammation. Number one is neutrophils. Neutrophils are most numerous and most important cellular component of the innate immune response. Neutrophils are typically circular in shape with a diameter ranging from 12 to 15 micrometers in humans. Activated neutrophils can change shape to extend pseudopodia for phagocytosis. They are characterized by a multi-lobed nucleus, that is 3 to 5 lobes connected by thin strands, and contain numerous granules in their cytoplasm. Neutrophils contain two main types of granules in their cytoplasm: azurophilic, that is primary granules, and secondary granules. Azurophilic granules, which appear purple, are rich in microbicidal substances and enzymes that help neutralize and eliminate pathogens. Secondary granules contain enzymes like lysozyme and collagenase, also contributing to the neutrophils' antimicrobial and tissue remodeling functions. Now, functions of neutrophils. Number one, phagocytosis. They are major phagocytes of the body with a primary role in the clearance of extracellular pathogens. Second is inflammation. Neutrophils are rapidly recruited to sites of infection or tissue damage, playing a crucial role in the early stages of inflammation. Antimicrobial peptides. They release antimicrobial peptides and reactive oxygen species to combat pathogens. Second cell is eosinophils. The eosinophil has a bilobed nucleus and a granulated cytoplasm that stains with the acidic dye eosine red, hence its name. Eosinophils are typically found in small numbers in the bloodstream, but they can migrate to tissue like the lungs, gastrointestinal tract and skin in response to inflammation or infection. They are chiefly involved in defense against parasitic infections because their number increase during a parasitic infection. Now, function of eosinophils. First is parasitic defense. Eosinophils are primary defenders against multicellular parasites like helminths, releasing toxic proteins and enzymes to kill or immobilize them. Allergic response. They release substances like histamine and other mediators during allergic reactions. Mast cell regulation. Eosinophils can interact with mast cells, influencing their activity through the release of cytokines. Inflammatory mediators. They produce cytokine mediators that attract other immune cells, enhancing inflammation and promoting tissue repair. T cell activation. Eosinophils secrete cytokines like interleukin 2, 4, 6, 12, which can stimulate T cell proliferation and differentiation. Now, the next cell is basophils. Basophils are a type of granulocytes that play a role in allergic reactions and inflammations. They are the least common type of leukocyte, comprising less than 1% of circulating blood cells. Basophils contain granules that store histamines and heparin, and they release these chemicals in response to various stimuli, including allergens and pathogens. Basophils are the least common type of granulocyte. Now, functions of basophils. 1. Allergic and inflammatory. They are involved in allergic reactions and inflammatory responses similar to eosinophils. They have a role in T helper type 2 response. Basophils secrete interleukins like interleukin 4 and interleukin 13. These cytokines promote the differentiation of naive helper T cells into T helper type 2 cells, which are involved in allergic reactions and defense against parasitic worms, helminths. Histamines and heparin. Basophils release histamines and heparins, which can increase blood flow and vessel permeability, contributing to inflammation. The release of heparin prevent blood from clotting in an area where an invading organism or allergen has caused damage. Antigen presentation. They can also participate in antigen presentation and differentiation of CD4 T cells. IgE production. Interleukin 4 and interleukin 13 also help B cells switch to producing IgE antibodies, which are important for fighting parasitic infections and are also involved in allergic reactions. Now, let us move on to mast cells. Now, mast cells are typically 10 to 18 micrometers in diameter with a round or oval shape. They have a single central round or oval nucleus. The cytoplasm is filled with numerous granules, which are characteristics of mast cells and contain various mediators. Mast cells are strategically located near blood vessels, nerves and epithelial surfaces like skin, lungs, gastrointestinal tract, allowing them to interact with various stimuli and participate in immune responses. There are two main types of mast cells: connective tissue mast cells found in skin and loose connective tissue, and mucosal mast cells found in the gastrointestinal and respiratory tracts. Now, functions of mast cells. Number 1, allergic reactions. Mast cells are crucial in type 1 hypersensitivity reactions. When an allergen binds to IgE antibody on the mast cell surface, it triggers degranulation, releasing histamines, prostaglandins and leukotrienes, which contribute to allergic symptoms like vasodilation, increased vascular permeability, itching and bronchoconstriction. Innate immunity. Mast cells act as sentinels detecting pathogens through pattern recognition receptors and releasing cytokines and chemokines to recruit other immune cells like neutrophils. Defense against parasites. Mast cells are effective in combating parasitic infections, particularly helminths, by releasing mediators and recruiting eosinophils. Wound healing and tissue repair. Mast cells play a role in tissue repair by releasing growth factor like VEGF and cytokines that promote new blood vessel formation and tissue remodeling. Angiogenesis. They secrete substances that promote the formation of new blood vessels, a process called angiogenesis. Now, let us move on to dendritic cells. The dendritic cells acquired its name because it is covered with long membrane extensions that resemble the dendrites of nerve cells. There are many types of dendritic cells, although most mature dendritic cells have the same major function, the presentation of antigen to T helper cells. 4 types of dendritic cells are known: Langerhans cells, interstitial dendritic cells, myeloid cells and lymphoid dendritic cells. They are most potent antigen presenting cells than macrophages and B cells, both of which need to be activated before they can function as antigen presenting cells. Immature or precursor form of each of these types of dendritic cells acquire antigen by phagocytosis or endocytosis. The antigen is processed and mature dendritic cells present it to T helper cells. Another type of dendritic cell, the follicular dendritic cell, does not arise in bone marrow and has a different function from the antigen presenting dendritic cells described earlier. Now, functions of dendritic cells. Dendritic cells act as a central link between the innate and adaptive immune systems. Dendritic cells capture antigens which are then processed into peptide fragments for presentation on MHC molecules. Dendritic cells are the most potent activators of T cells. Dendritic cells secrete cytokines that activate T cells, stimulate natural killer cells, enhance inflammation and regulate other immune cells like B cells, macrophages, etc. Plasmacytoid dendritic cells detect viral infections and produce large amounts of type 1 interferons, which inhibit viral replication, activate natural killer cells and enhance antigen presentation. Now, dear students, I hope you understood today's lecture. See you next time. Thank you.